Welcome everyone. We should be live on YouTube and live on Zoom. Welcome to the Scottish final of Famelab. I'm Jamie Gallagher and I will be guiding you through tonight's proceedings. And we've got a selection box of science in store for you. We have 7 fantastic contestants who have just 3 minutes to share some interesting science with us. On board, we also have 3 judges who are going to watch the talks and then interrogate the contestants straight away afterwards. If you are not familiar with Famelab, I'll give you just a little bit of a background as to what the competition is all about. It is an international competition that has been going for over 10 years, and it is a search for new science communication talent. Contestants, as I said, have just 3 minutes to share some interesting science, technology, engineering, maths, or medicine. And they're allowed props, but crucially, no PowerPoint slides. That is one very, very strict Famelab rule. No PowerPoint in this competition. Contestants will be judged on 3 different criteria: on content, clarity, and charisma. So that's what the judges are going to be looking out for. At home, please do let us know what you think. So if you're watching on YouTube, you'll be able to write down in the comments. If you're watching on Zoom as well, there's that little text box. So let us know what you're thinking, where you're watching from, and I hope you enjoy tonight's show. So the first thing I want to do is introduce our 3 judges. So the first judge that I'm going to come to is Suzanne. Suzanne is based in Glasgow, but lives in Edinburgh, so giving us that kind of Scottish central belt wisdom. So first of all, I want to say hello to Suzanne. And could you just introduce yourself a little bit and say what you'll be looking for from our contestants tonight? Suzanne.
Hi, good evening, everyone. So my name is Suzanne Mcadoo. I'm the Public Engagement Coordinator in University of Strathclyde over in Glasgow, but I am coming to you from my bedroom in Edinburgh. I'm really looking forward to tonight. And what I want to know is I want to be watching for the person who I want to, after all this is over, take out to the pub and really learn more. That's really the question I want answered. Who am I taking to the pub after we're all out of our houses?
Yeah, so a bit of a delay there. But yeah, so eventually, Suzanne, that might be part of the prize that you're winning, a night out with Suzanne in a pub. We might hold you to that. Next judge that we're going to come to, Tony, could we come to you and say a little bit about who you are and what you might be looking for from our contestants tonight? Tony.
I'm Tony Weir. I'm from Howell University. So a long time ago, I was a researcher and talked about squirrels. So if anybody can slip in squirrels, that would obviously work for me, but it's not obligatory. But now I'm at Howell University. I'm head of policy, strategy and impact there. So what I'm looking for is people who can convince me that their work is interesting and it's going to make a difference.
Okay, brilliant. Thank you, Tony. So interesting and making a difference. We want to see a couple of different sides to your research. Our third judge, and we're going to come on and again ask you to introduce yourself and say a little bit about what you're looking for. Mary, could you introduce yourself? Tell us who you are.
Yeah. Hi, so my name's Mary Irwin. I'm an honorary research fellow in media and communication at Queen Margaret University. My background is arts and humanities, so I'm looking for you guys, 1, to present something that I can understand, and number 2, get me fired up about science and technology.
Excellent. Thank you. Thank you, Mary. So arts and humanities background, because this is a competition for everyone. This is not for specialist audiences. This is a selection box of science for everyone, science fans and non-science fans yet. A little bit more about the competition. Just to let you know, this is a competition that was started off at Cheltenham Science Festival. You can see their logo just up here. Cheltenham Science Festival is not kind of taking place this year because it's in June, so it's had to be cancelled, but Famelab competition is still running. It's a competition that takes place all around the world, and all around the world just now, there are finals like this taking place. Tonight, we're going to be looking for 2 winners, and these 2 winners will go forward into the UK final of Famelab, where we'll be looking for just 1 contestant to represent the UK at the international final. And as I said, countries all around the world just now looking for their international representative of science communication. And so tonight, we're going to be looking for our 2 champions to represent Scotland in that crucial UK final, which again will be a virtual final. I should just say that normally Famelab is a live event, takes place on a stage. Our judges are very well aware of that, and they have agreed to take into consideration the limitations of Zoom. So this is going to be a Famelab final with a bit of a difference, but I'm expecting no less clarity, content, and charisma from our contestants. So without further ado, we are going to jump straight in to our first contestant. I'm going to get my timer ready. And our first contestant is Carol Chermas. Carol, hello, and off you go.
Hello, everyone out there listening to me with your headphones on in your bedroom. Not quite the scenario you had in mind for Famelab, maybe. Well, these days we seldom speak in person to someone else. We have Easter lunch on Skype and birthday parties on Zoom. Cheers. How many times have you done that in the past month? All the human speech that we listen to comes out from devices. And this is why I feel that my research is really on spot now. And I'll tell you what I do. I study ways of making speech signals more intelligible. So for example, think of when you're trying to listen to the radio on the loudspeaker, but you're cooking, you're frying something in the pan with the fan on and it goes, and you're trying to focus really hard on the speaker, but you can't because it's loud. And so you would like to raise the volume, but you don't touch it because your hands are dirty. And even if you do, your partner in the other room is going to go, will you turn that thing down? So wouldn't it be nice if there was a sound engineer in your kitchen adjusting the sound to your radio so that you wouldn't have to raise the volume? Well, there is no sound engineer in your kitchen, well, usually, but maybe it doesn't have to be a person. Maybe it can be an algorithm, and we have developed one. Did you know that the most important frequencies for humans to understand speech are between 1 and 4 kilohertz, but actually most of the energy in speech signals are below 1 kilohertz? And did you know that consonants, the sounds like t, sh, ch, are key to understanding speech, but most of the energy is found in the vowels like a, o. So what we do is to reallocate the energy. We optimize the signal without raising the volume. So we tested our algorithm in 3 different languages, and we found that in the best case, it improved the intelligibility of speech in noise from 15% to 73%, which means that instead of getting 1 or 2 words out of 10, you'd get 7 or 8. Why is this important? In the UK, 1 in 6 people are affected by some degree of hearing loss. And most of those who could benefit from the use of prosthetic devices do not, because they feel uneasy at the idea of asking for help. So our technology can help to some extent, but it is not a substitute for medical help. So if you feel that something isn't quite right, don't be afraid to ask your doctor. 3 years ago, I left everything that I had to pursue my research. I'm not afraid of making big, scary changes. Now, are you? Thanks.
Brilliant. Thank you, Carol. A sound engineer in our kitchen. We could all do with that, especially with the amount of Zoom calls that we're all dealing with nowadays. So half of your ordeal is over. Thank you so much for sharing that. But now we have the judges' questions. So judges, do you have any questions for our sound engineer?
Yes. Am I spotlighted? First of all, cheers. Really interesting. I loved how you made it quite relevant to what we're all experiencing right now. You talked about kind of the ch and sh versus the vowel sounds. So would this be different for different languages in terms of like the frequency or what combinations of sounds? Would solving this problem have to be kind of regionalized for different languages or language groups?
So not really, because consonants and vowels are not specific to any language. I mean, we have the sounds in every language. So some languages are more oriented, are more full of vowels, like Italian, for example, whereas like German or English have more consonants. So this is true, but in both languages, consonants are very important for understanding speech. And one interesting thing about different languages would be trying this algorithm on tonal languages. So like Chinese, for example. We believe that it will work the same, but we have to test it. So stay tuned for that.
Brilliant. Thank you.
Brilliant. Thank you, Suzanne. Any of our other judges have a burning question to put to Carol?
I have Jamie.
Yeah, please.
Yeah. How significant, Carol, do you think the work that you're doing is? How important is it?
Well, I feel that it is very important because, as I said, there's many people, not just in the UK, but all around the world, that do not, they don't express the need of using prosthetic devices or asking help in general when they feel that their hearing is not right. And I know many people that think that it's just not severe enough, or they are afraid of the stigma of wearing prosthetic devices. So I think that at least if technology can reach out to these people, which are actually the majority, because the majority of people who have some degree of hearing loss do not ask for help. So I think that if we can reach out, if we can get this technology in different devices like radio or headphones or mobiles, then we can help to some extent. And I know that I would like to have this technology in different devices, even though I am apparently normal hearing.
Thank you very much.
Brilliant. Thank you. We've got 30 seconds if you've got a burning question, Tony, or are you happy?
No, I'm happy. My question actually was the same as Suzanne, so I don't need to ask it.
Brilliant. Well, thank you very much, Carl. Let's give Carl a round of applause. Yay. Excellent. Thank you. Thank you. So that's our first contestant. One of the most challenging tasks is to go first, but you did it fantastically. And we are still live streaming on Zoom and on YouTube. Comments coming in from both of those sides. So it's really, really nice to see. Hello, everyone that's watching at home. Thank you for joining in. I hope you're paying attention because there will be a quiz later on. Literally, like, pay attention. But we've got our second contestant coming up now. We're going to move on to Richard next. So Richard, you are getting ready. You're going to take stage for your 3 minutes. Give us something interesting, give us something clear, give us some content, and give us some charisma. Hello, Richard.
Hi. So, playing music to dogs has the potential to reduce anxiety and improve welfare. But generally, the effects are quite modest. Working with the Scottish SPCA, we have found a really simple and effective way to strengthen the calming effects of music. For some human patients, these tablets will provide as much pain relief as morphine. However, these are way, way, way safer than morphine. They're way safer than morphine because they're just sugar tablets. They are a placebo. Now, placebo doesn't mean made up. The placebo effect is a genuine body systems response to something that shouldn't be having a body systems effect. But crucially, it depends on an expectation of benefit on the part of the patient. Now, even real drugs have some aspect of placebo effect, and therapies work far better if the patient believes they're going to work. Now, of course, you might say this is all just in your head. Well, the truth of pain or anxiety is that although the inputs may be very, very real, the way those signals are processed by the brain is quite literally all in your head. Now, I know what you're thinking. You're thinking, wow, this is really cool, but what's it got to do with music and dogs? Well, in human studies of anxiety, at least some of the benefit from music comes from an aspect of placebo effect. And we know this because the greatest benefit comes when patients can select their own calming music. They already know they find this music relaxing. So there's already that expectation of benefit. Now, of course, the problem with dogs is they don't know that this music is supposed to be relaxing. So I think we can probably all agree that there can't be an element of placebo effect. But I think we might be wrong, except with dogs, we call it training. What we've done is to give a number of dogs meaning to a piece of music. We've taught the dogs that a particular piece of music means something. We've played the same piece of music at the rescue and rehoming centres every night at bedtime, so the dogs associate it with going to sleep. We've given them that expectation of benefit, and it works. when we play the same music during the day, it has a stronger calming effect than it does before the training started. Now, this has all been done at the rescue and rehoming centres, but there's absolutely no reason why we can't train our own pets at home to have a go-to piece of music for times of increased stress, such as bonfire night or thunderstorms, because we already know that music works to reduce anxiety and improve welfare. We now know how to make it work better.
Brilliant. Thank you, Richard. That was brilliant and bang on time as well. So I mean, my issue is that when I was at school and doing science, no one told me that doing science with dogs and playing music to them was a subject. And that's my own axe to grind that I'll deal with later, my guidance counsellor. But first of all, let's go to our judges with some questions for Richard. So judges, who's going to kick us off with some questions?
Can I start off, please?
Please do, yeah.
So Richard, I'd like to know what got you into this line of research.
Well, my background was really stress and welfare, actually in horses. And I became very adept at measuring stress and quantifying stress. This PhD came up funded by the SSPCA, who were interested in basically environmental enrichment within the kennels. And they thought auditory enrichment was a good target because it's simple, it's cheap, it's relatively effective. So, I swung across, from playing with stress in horses to looking at mitigating stress in dogs. It was a bit of a struggle of a transition, but not too difficult.
It's very helpful. Thank you.
Brilliant. Thank you, Mary. Any other questions? Tony, come to you.
So I have a question about whether you think there's any sort of limits to it. I mean, I can imagine maybe a few other mammals being, benefiting in the same way, but would it extend to the same level for people who keep stick insects as pets? Is there what range of pets do you think this might cover?
Well, there's been a lot of work done with birds, particularly songbirds. Obviously, for them, auditory enrichment is really important. I don't know is the simple answer. I suppose the question is, how would you measure it? I mean, how do you know when your stick insect's stressed or relaxed? That would be the problem. But I think for a lot of animals, the biggest part of it is the association they're making. So if you can associate a sound and give it meaning to something that's relaxing or exciting or stimulating, it doesn't really matter on the species. So if you could probably train stick insects to respond to auditory stimuli. So if they could associate it with something relaxing, it'd be great.
Okay.
Brilliant. Stressed stick insects. Suzanne, there's a little bit of time if you've got a question.
I'm not sure how to follow stressed stick insects, but is there any kind of type of music that tends to be particularly good for this kind of training, or is it just as long as the training is there, it doesn't matter so much?
Well, I mean, there are two aspects to it. I mean, dogs' hearing is more sensitive than ours, but their sensitive range is about the same. So really music, we're listening to as far as pitch and volume is concerned, it's about the same as what we find relaxing, they'd find relaxing. The key to it really is not rapid changes. So no rapid key changes, no rapid changes in volume. So the things that fit that, reggae works quite well, certain classical music works well, Mozart's quite good, 1812 overture, less so. Because there's crashes and bangs in it. So rather than a genre music, it's just looking for stuff that's, doesn't change too much. Although there is work that shows that heavy metal music isn't so good.
Brilliant. So that is the joy of Famelab. I think that we're all learning tonight. And if you have a dog at home, then get that reggae music out and relax your dog. If you've got some stick insects, there's the project just waiting to happen. Is it stressed? Is it happy? Who knows? Try the reggae music. Try Mozart. Try something else. Maybe stick insects like pop. We might never know unless you go and experiment. But anyway, thank you very much for Richard. Let's have a round of applause for Richard Mott. Yay. I realize people at home probably can't see, but the other contestants are clapping. I can see the other contestants, they're clapping. It's not just me, I promise you. We're a loving, supportive family, is Famelab. We've got another contestant, and we're going to move straight on now. Our next contestant is Jessie Howell, and again, we're in store for an absolute treat. Jessie, can you say hello to us and give us your 3 minutes of wonder?
Hello. So, genetic engineering has always been a controversial topic. We've all heard about the dark side of manipulating DNA, like making designer babies, cloning extinct animals, or making animal-human hybrids. And we've seen the results in sci-fi movies like Jurassic Park or Splice, which is a terrifying movie if you haven't seen it. But on the flip side, genetic engineering is also an incredibly powerful tool, which can be used in industry, research, and it can, we can use it to design super crops to feed the hungry, or design trees which can reduce pollution, design bacteria which can mass produce drugs like insulin. In my lab, we are using it to aid studies into the horrible parasite leishmania. This parasite affects millions of people worldwide, causing sores, disfigurements, and even death, killing around 70,000 people every year. With very few treatments and no vaccines. In my lab, we are looking at how these parasites replicate. When they do this, parasites need to replicate in order to survive. And when they do this, they form distinct populations with differences in their shapes and sizes. Now, if we can separate these parasites based on these distinct shapes and sizes, then we have the potential to look at replication-specific proteins, which may be targetable with drugs and provide possible new treatments. Yet these parasites are 10 times smaller than the width of the human hair, making seeing them, never mind separating them, incredibly difficult. Genetic engineering can make this much easier. I can make my parasites glow and light up like a Christmas tree. Using fluorescence makes determining the size and shapes of these parasites much easier, and therefore making them easier to separate. So how do I make my parasites glow? Well, as it turns out, thousands of organisms in the ocean fluoresce naturally. And from these, we have derived hundreds of different fluorescent genes, which produce many different colors. So I can transfer DNA from these bizarre marine organisms into my parasites. And voila, Christmas tree-worthy parasites. And if that isn't impressive enough, you can actually do this sort of experiment in your own home. Not with killer parasites, that would be dangerous. But you can actually buy kits online to make your own fluorescent yeast. A fun isolation activity? So yes, genetic engineering has to be used sensibly, but I'm hoping to use this to study leishmania in the hope of finding a cure. Thank you.
Yeah, brilliant. Thank you very much for that. Fluorescent yeast. I mean, I don't know if that's easier or not to find than normal yeast, given the current situation. But I don't get to ask the questions. Sadly, it's up to the judges who get to ask their question. But judges, that'd be a good question. Just saying. So let's move on to our judges now, and let's go to Tony. Tony, do you have a question about the parasites, the fluorescence?
So you mentioned a few films with genetic engineering in. I just wondered, which is your favourite representation of genetic engineering in a film?
Oh, I did really enjoy Splice, as terrifying as it was. That's making human-animal hybrids, and they were showing the different possibilities. They were showing animals which were mating and animals which became very clever and human-like. So that was probably my favorite, but not something I want to see in the future.
I just wondered if it was Toy Story, because all of the props that you were showing up reminded me a bit of the toys in the next garden.
Yeah.
Thank you.
Brilliant. Thank you, Tony. Any questions from Mary or Suzanne?
I'd like to ask Jessie, how can you make the public less anxious about genetic engineering? How can you make non-scientists feel less scared or less worried?
I think it's more about the awareness of it. I mean, in the US, they already have a lot of food which is produced by genetic engineering, which isn't causing any difficulties or impacts as we've seen. But also the amount of drugs and things and the treatments available for gene therapies, for example, all of these would be so beneficial. So it's just about raising awareness of how it can be used.
So I guess it's getting information out to the public better, and about things that they're interested in that they didn't already know were genetically engineered. Thank you.
Brilliant, thank you. Suzanne, there's a little bit of time left if you've got any questions.
Okay, so you say you managed to get like you had a kind of a long boy and a square boy, and you get them to fluoresce in the way you want them to. So what will be the next step for this research?
So we're using their fluorescence to separate them based on their size and shape. So using the fluorescence, we can see the different shapes. And from this, with the different populations, we can identify replication-specific proteins, which are imperative to their survival. So if we can block those proteins, then we might be able to find a treatment or a cure for the parasite.
Okay, thank you.
Brilliant. Thank you, Jessie. A massive round of applause for Jessie, everyone. Yay. Brilliant. Well done. If you are, like me, a massive fluorescence fan, you can go on Twitter, and Fluorescence Friday is a thing. I honestly, go and look this up later after Famelab. Go and look up Fluorescence Friday, and chemists, biologists share the beauty that is fluorescence. While we're on a Twitter tangent, do also make sure that you're tweeting along and sharing what is happening here. You can find Famelab just at Famelab. You'll find them on Twitter, and there's lots of activities going on. Follow Famelab, because then you'll get to see what is happening, not just tonight, but as we go forward into the UK competition and then through into the international rounds as well. So get tweeting and sharing what is happening here tonight. So thank you very much to Jessie. We're going to move on to our next contestant now, rattling through them from one area of science to the next. And our next contestant is Carlin Sound. Do you want to say hello? And off you go.
Hi. So we love to believe that all of the technology around us is smart. We all have smartphones, smart watches, maybe some of us have smart TVs. And the way that this technology proves that it's smart is basically just by making predictions. YouTube knows what you want to watch before you do. Autocorrect knows that when I type, "How are you?" I'm probably going to type "doing" next. And this predictive power comes from a family of algorithms called neural networks. And the phrase neural network is tossed around a lot when we talk about artificial intelligence, but here's the essence of how it works. You have something that you want the computer to understand, say a picture. You want the computer to guess if the picture has a dog or a cat, to give it a label. And in this case, each individual pixel in the image acts as an input, and the output is how sure the computer is that the picture's of a dog or a cat. And the computer can be, say, 80% sure that the picture's of a dog and 40% sure that it's of a cat. This guess is unrestricted. So the way the algorithm works is that in between the input and output, there's what's called hidden layers of this network that are made up of individual nodes. And this means that each of these individual nodes is making a decision about one little piece of the input. For example, is the pixel brown? Yes or no? But importantly, we don't know the calculation exactly that the node is making. But then each subsequent layer makes a decision about the previous layer, on and on until you get to your final output. And then in the output is where the training happens. You get to see how right you were. If there was a dog in the picture, did the computer guess it? Then you take the difference between the correct answer, yes, there was a dog, and your computer's guess, I was only 60% sure there was a dog. And based on that difference, you make tiny adjustments to each of the nodes that fed into the answer, so your next guess is slightly more accurate. And when you do this about 10,000 times, you normally have a pretty accurate dog or cat guesser. And believe it or not, this is the essence of how nearly all of our smart technology actually works today. So we can take areas that have a lot of labeled examples and train neural networks to learn patterns in them. And this is important because computers don't get bored sifting through page after page after page of medical journals, and they don't get tired from hours of driving looking at the road. So we, as humans, with our limited capacity and our limited thinking, can't see the patterns that they can see. By having neural networks that help us process all this information around us and making our own decisions that are guided by those suggestions, we can build a safer, more advanced, and smarter world. Thanks.
Brilliant. Thank you. Excellent. Yay. More dogs. Famelab double dogs. Excellent. I'm loving it. I have got so many questions, but I'm not allowed to ask the questions. So we're going to go straight on to our judges and see if the judges have any burning questions.
If I may start. So that was really I was quite interesting, because you say this is kind of a technology we're dealing with a lot unknowingly. What do you think are the ethics around this when you don't know how the computer is making the decisions? Because we've seen cases where algorithms that are trained on just a subset of photos of the human population are unable to deal with differences in race, for example. So what are the ethics of using this kind of technology from your point of view?
Totally. So that's a really good example. So affective computing is an area that deals specifically with emotion and deals with these tools, neural nets. And one thing that's very hot there right now is trying to guess if someone is lying or not. And long story short, we can't. Neural nets can't do that because humans can't do that. And I think where I fall on the ethics is if you have a computer that's trying to make a decision about another person, another human needs to be really heavily involved in that process. Lots of folks in AI think will go even further and say computers should not be able to make any decisions about what happens to a person or about people. But of course, there are also cases where driving, for example, your computer is making decisions about people insofar as it's keeping people safe, but it's doing that in an objective way, in a way that can be objective. So I guess the short answer to your question, I would say is, if you are trying to learn something about a human, there need to be other humans really heavily involved in that process, and ideally also a pretty large ethical overview with public conversation.
Thanks.
Brilliant. So any other questions from our judges, Mary or Tony?
Can I ask, how important is specific information about gender in terms of building up data in the kind of work that you're doing in building neural networks?
So it really depends on your application, right? A neural network is a tool that can be used for anything, but I'll take voice as an example that we had earlier tonight with Famelab. So if you're creating an algorithm that you're trying to train to recognize voices, you basically just need to make sure you have a wide sampling of voices. Early voice recognition algorithms didn't work on women, or actually famously did not work on Scottish accents either. So I think, yeah. So I think that it just depends on your application, right? If you're deciding pictures of dogs or cats, then gender doesn't really play a role there. But I think with all things with neural networks, your application domain is the most important thing. Where are you trying to insert this tool? And do you have an expert who can see all the sides of that?
Great, thank you.
Brilliant, thank you for that. Tony, did you have any questions or are you happy?
I've got one question. You talked about a succession of layers with one question. How many layers are you talking about? Is this sort of just a few or thousands, or how deep does it go?
Yeah, so that is something that machine learning is always trying to do. Basically, you can optimize to have the best number of layers that gets whatever your specific application wants in terms of a trade-off between space, speed of training, accuracy of response. But there's not really a limit. You could have just one hidden layer in there. You could have 5 hidden layers, but each one has 300 nodes in it. They all achieve different things and are sort of designed for different specific applications.
Thank you.
Brilliant. Thank you for that. So another trio of questions there. By the way, if you're watching at home and you're thinking about neural nets and how neural nets need to be trained to work out whether it's a dog or a cat, just letting you know, I'm brilliant at deciding whether it's a dog or a cat. So if you want to send me photos and ask me, is this a dog or a cat? I am willing to do that public service. We do not need to involve the computers. No AI, no Rise of the Terminator stuff. Just send all your dogs and cats to me and I'll tell you if it's a dog or a cat. I'm willing to make that sacrifice for you. So we're halfway through or over because we've got 7, which we don't divide the speakers in half. So we've had 4 of our speakers. We still have 3 more to go. If you're watching at home, again, just welcome. There's lots of people watching on YouTube. Thanks for all the little waves. We've got Nate and Stephanie and Sandra, hello, and comments on Zoom as well. So hello to Zara, lots of people watching at home, which is lovely. Keep cheering those contestants on. It's really nice to see the support coming from the internet as well. So thank you for that. Okay, I'm sorry, I keep getting distracted because I can see myself on a slight delay. So when I wave at myself 5 seconds later. That's quite distracting. But I'm with you, I'm concentrating, promise. This is me being professional. So we're going to move on to our next contestant now. Our next contestant, again, competing for that spot, looking for our 2 winners that will go through to represent Scotland in the UK final. So our next contestant to share 3 minutes of interesting science, technology, engineering, maths, or medicine is Elizabeth Mills. Elizabeth, hello, take it away.
Hi. Today, I want you guys to come with me on my absolute favourite activity. Put your wellies on, we're going rock crawling. Are they on? Imagine that you are traipsing around your local beach or maybe somewhere you explored as a kid. Walking over the seaweed, don't slip. As you reach into the cold water, lift up a rock and see barnacles, anemones, seaweeds, all growing on the rock's surface. Now, these species started their lives off as tiny little larvae that you can barely see with your eye, if at all, that then decided to settle and live on the bottom of that rock, where they metamorphosized like a caterpillar into a butterfly, except when these guys turn into adults, they are never going to move from that spot again. Now, excuse me as I wrench you from your own childhood memories and plop you in one of mine. Instead of a rocky shore, we're going to be exploring a sunken shipwreck when the tide is out. This metallic ship was left there since the World Wars, but now nature has taken over. And like our rocky shore, it's full of slippery, slimy, and squelchy marine life. But if you look closer, actually some of the species you see on this ship are different to the ones you found in your favourite rock pool. And some of the ones you found aren't on there. Why is that? Well, when you've only got one place to pick and live for the rest of your life, you're going to become fussy. And that's what my research and other researchers has shown, that at this larval stage, they have the ability to determine between different materials. They can tell if it's metal or stone. And this means that they also have favourite places to set up their homes. And when you look at man-made surfaces, you can find different species than on natural ones. But that's led to a little tricky problem that I'm trying to solve. On man-made devices, we often find species that have hitchhiked on the bottom of ships around the world that set up home in the UK. That becomes a problem when these species don't play nice with our native sea life. In fact, they can threaten our rock crawling communities. Now, as you can tell, I like wearing hats, and in my career, I have worn many hats. I'm a marine biologist, an engineer, and now I am a rocky shore real estate agent. And that's because I design new materials for and new homes for these creatures, so that when we put them in the sea, the creatures that threaten our rocky shore communities don't actually want to set up home anymore. I started my days as a kid by putting on my wellies and asking curious questions about marine life. And my adult days start pretty much the same way, except now what I'm finding out is actually helping to save and protect the rocky shore creatures I love so much. Thank you.
Brilliant. Thank you, Elizabeth. Well done. Yeah. Rock pool communities. What a beautiful phrase. Like, I'm living out a whole series that needs to be on TV of rock pool communities, and I'm imagining little friendly crabs and anemones that get on really well together, which is such a beautiful image in my head. So thank you for that, and thank you for the themed hats. I'm all here for the themed hats as well. Fantastic contribution to Famelab. Because again, you can use any props you like. If your prop is a hat, then, you know, bring on the hat themes. Okay, so judges, is over to you now. Rock pool communities, questions, please.
I'd like to ask Elizabeth, how would you engage the general public with all this fantastic work that you're doing?
I think a very simple statement is just all you need is your wellies for science. Good. It's something that people do as a kid, but it's not just for that, it's for adults as well. And telling people that science is as simple as putting on a pair of wellies, I found really excites people. They don't realize how much we have right on our doorstep, right, with easy access. There's no, marine biology isn't fancy scuba diving, it's not, you know, traveling around the world. Sometimes it is just putting on a pair of shoes and asking curious questions.
So it's like breaking down barriers and making it clear that you don't have to have test tubes or algorithms, you can just go outside. Thank you, great.
Brilliant, thank you, Mary. Any other questions from our judges?
If I can, what's the most interesting thing you found walking along the shoreline?
I found down the road at Anstruther in Scotland, I found a little nudibranch, which is only about this size, but it's a tiny, delicate sea slug that are really quite rare. And it was really tiny and really, it was just gorgeous to see and spot. And if you have a look at nudibranchs online, they are multicolored sea slugs that eat hydroids and become poisonous, and they're just these weird and amazing creatures.
Thank you.
Brilliant, thank you. Suzanne, anything from you?
I was going to ask what your favorite was, but I think you may have just answered that. But you said that a lot of these creatures can kind of tell what kind of surfaces they're on, like wood, metal, rock. So what kind of senses did they use to figure that out? I'm
not actually sure exactly what the senses are. We're mainly going from testing experiments where we give them options and then they pick. But a particularly great one is a barnacle has these two basically feelers on the front of their head. It will feel along, and if it likes it, it will put like a post-it note amount of glue down and see if it likes it. And if it doesn't, it hops off, and you can follow the footprints of the post-it note glue. And eventually, when it does settle, it releases one of the strongest glues in the entire world and will never, ever move again. So that's one of the species we know more about, but I'm not entirely sure what senses these teeny, weeny little things actually using.
Cool, thank you.
Beautiful, thank you. And what a beautiful sentiment that wellies are all you need for science. I mean, I guess it depends on your science, but it's a beautiful sentiment. As far as my marine biology goes, it really stops at poking dead jellyfish with sticks. That's kind of as far as I went. But now I'm more curious. I'm curious to walk along the Clyde and see what I can find. I know there's things in there, maybe nothing terribly exciting or beautiful, but you've convinced me to go and find some little seaside communities. I would say one of the comments that's come in on Zoom from Susan, who many of you will know from Cali, organising lots of community and public engagement over there. Her comment goes to the judges as well, often neglected in this process. It's an incredibly difficult job to be a contestant, but also a really difficult job to be a judge. The questions are coming thick and fast. They're having to make split-second decisions on this. And Susan was saying that the judges are doing a fantastic job with the questions, that she knows it's not easy. And this is a great way of relaxing the speakers. So praise coming in there for the judges from people watching at home as well. And also, Carlin, I'm thinking, is this your mum maybe is watching, or your sister, or someone saying, yay, Carol, which is so cute. So Carlin, I think someone there is cheering you on as well. And we're getting still lots of waves and things like that coming in as well. So hello to everyone that is watching on YouTube and on Zoom. Thank you so much for joining us. And again, pay attention, because after our final speaker, which is coming up soon, there's going to be a quiz while the judges make that really difficult decision. So we have just 2 contestants remaining now. And so we're going to move on to contestant number 6 for 3 minutes of interesting science, technology, engineering, maths, or medicine. Let's say hello to Anna. Anna, hello.
Hello. Can you see me?
We can. Off you go.
Great. Who remembers playing the mystery box game as a child? You would sit together with your friends at Halloween parties and pass around a mysterious box. You would reach through a small cutout and try to guess the spooky objects inside. Are these pipe cleaners or spider legs? Am I touching mini marshmallows or ghost poop? As neuroscientists, we face the same issue. We want to know more about the brain, which is protected by a secure box made up of our skin, skull, and meninges. Of course, it's not feasible on a day-to-day basis to make cutouts, so we had to come up with a different solution. And the answer is light. The name of this technology is functional near-infrared spectroscopy, a deeply boring name for an incredibly exciting thing. And spectroscopy is the science that looks at how light interacts with matter. The functional bit refers to brain activity rather than brain anatomy. And the near-infrared part describes which type of light is used. With this, which type of light is used? The visible light spectrum contains all the colors of the rainbow, each with different wavelengths. Maybe you have noticed that when you shine a flashlight directly onto your hand, it emanates a red glow. This is because colors with shorter wavelengths like green and blue are scattered or absorbed. Red gets scattered and absorbed the least, and this way it can make the mystery box surrounding the brain transparent. With this brain imaging technique, we place mini flashlights called optodes onto the head and place detectors next to them. Because we know how red light interacts with biological tissue, we know that the path of the light will follow a banana-shaped curve through the outer layers of the head. Now, the final ingredient to making this scientific magic trick happen is down to a key property of our brain. Blood is oxygenated more if a brain region is engaged more. Lucky for us, oxygenated and deoxygenated blood absorb red light to a different extent. We can measure this difference with our detectors and boom, we get brain activity. The main advantage of using this brain imaging technique over others is that it allows us to do something pretty futuristic. Mobile brain imaging. People can put the device on their backs, walk around, and even talk to other people. This gives us incredible new insights into our brain's function, especially when it comes to socially interacting with other people. To this day, we still play the mystery box game in human brain imaging. The data we obtain is messy and imperfect. However, maybe because of this, we have retained our childlike wonder and curiosity for what really happens inside the mystery box. Thank you.
Yeah, brilliant. Thank you. Well done, Anna. Magnificent. If you're playing some sort of Famelab drinking game at home, that's a prop. Take a drink and an amazing reveal as well. That's 2 drinks. Finish your drink. There is no Famelab drinking game, but that's something that will eventually come to be about because it should. Famelab drinking game next year. So thank you for that. Again, I'm not allowed to ask any questions. Our judges have their drinks. Apparently, it's a mug though, so I think we're fine. Judges, we're going to come over to you and see what questions you have for Anna in that fantastic talk.
Yeah, so it's just coffee. With some of these imaging techniques, it can sort of take a long time after you've put the person through the whatever the machine is before you get results that you can analyze. How long does it take in the technology that you're developing? How quickly do you get results that you can use?
Oh, right. So I can't claim the fame of developing the functional near-infrared spectroscopy all by myself. I'm just using it at the moment. So it doesn't take quite as long as putting people, for example, in an fMRI. So we can simply put on the cap, we attach the optodes to the cap and the detectors as well. And that usually takes us between 10 and 20 minutes. And then people wear the cap between 20 minutes and half an hour while they interact with our robots. And so we're trying to understand more about what these embodied interactions with the robots can teach us about our brain's function.
Okay, thank you.
Brilliant. Any other questions from our judges, Suzanne or Mary?
Okay. What are the best strategies for explaining complicated science of the brain to people who don't really know very much science? It's a huge question, but I'm just interested.
I'm sorry, can you repeat the beginning?
Yeah.
I couldn't hear that.
Yeah. So what are good strategies for explaining the kind of work you're doing on the brain to people who don't have very much scientific knowledge?
All right. I think the key ingredient is, maybe it's a bit cheesy, but like enthusiasm. I think any neuroscientist that you meet on the street is still super in awe of the brain. And I think because there's just so much incredible things that we have yet to learn, that enthusiasm hopefully carries over to the person that you're trying to explain the research to.
It's a great answer. Thank you.
Brilliant. Suzanne, did you have anything?
Well, just so I think you mentioned infrared was the light you were using. I mean, are there any like safety concerns with this, or like how would it compare in terms of safety to other, more involved imaging that we might find in a hospital?
Yeah, that's a great question. So actually this brain imaging technique is one of the safest that there is, and it's really one of the least invasive ones. In fact, it was developed to monitor young babies' brain function. So yeah, it's using near infrared light. You can't feel it. The only restriction is to wearing the cap that gets uncomfortable at some point, but indeed it compares in terms of invasiveness and portability and also affordability. It really wins in comparison to many other brain imaging techniques.
Thank you.
Brilliant. Thank you. And another round of applause for Anna. Well done. So fantastic. We have taken a tour through brains and marine biology and dogs and a whole range of subjects. We have just one more Famelab talk to go. What's going to happen after this next Famelab talk is our judges will have to make a decision. They're going to have to work out who should win. And Mary was asking about excellent ways of communicating science. And if you're a Famelab fan, you will know that content, clarity, and charisma are three very good ways of communicating science, and that's what our contestants are being judged on tonight. So after our final contestant, we are going to send the judges away. We'll have a bit of a quiz, but right now we have one more contestant. Last chance to find that special place to be one of the two UK finalists. Our last contestant is Trisha Palmer. Hello, off you go, Trisha.
As a medical doctor and now a master's student in immunology and inflammatory diseases, I've come to realize that nothing is as it seems. So ladies and gentlemen, I present myself to you, the prop, and this is my story. One word, 4 letters, a word that incites so much emotion. And no, I do not speak about love, lust, or even fame. I speak about pain, neuropathic pain in multiple sclerosis. 2.5 million people are affected by multiple sclerosis globally, and it is a disease whose underlying cause remains unknown. We know that multiple sclerosis causes damage to the insulating layer of nerves that cause an abnormal crosstalk between nerve and nerve. And it is this abnormal electrical conductivity that results in neuropathic pain. Neuropathic pain affects 8% of the general population, but this number escalates to 50 to 86% of patients with multiple sclerosis. This causes a decay in the quality of life of these patients, because not only do they have to contend with neuropathic pain, they also become fatigued, depressed by multiple sclerosis itself. Now, you would think that having neuropathic pain is bad enough, but this pain presents in several different forms. It can present as paroxysmal, shooting, lancinating pain in the face and in the jaw that is diagnosed as trigeminal neuralgia. Or it can tickle the spine with electrical shock-like sensation radiating into the legs, diagnosed as the Lurite phenomenon. Or in some patients, it can present as muscle spasms. But for 41% of multiple sclerosis patients, sadly, they face severe tingling and burning and cramping sensation in the feet that keeps them awake. So, now that I've told you all of this, think for a few seconds that those legs burning in unquenchable pain is yours and not mine. So my question to you, ladies and gents, is how could you cope with a lifelong diagnosis of neuropathic pain? Or better yet, could you even cope at all? Thank you.
Brilliant. Thank you. Round of applause for Trisha. Well done. Excellent. So another impassioned Famelab talk, our last Famelab talk of the night, but the night isn't over yet because the next thing that we're going to do is have questions from our judges. So judges, do you have any questions on pain?
Questions on pain. That's I mean, you really kind of portrayed it quite evocatively, and I hesitate to say beautifully, but I think quite beautifully there. But obviously this is, I mean, getting rid of this pain, I assume, would be kind of an end goal. Is there kind of an idea of how people might kind of deal with this issue? Is there, I guess, kind of like ibuprofen isn't going to help, but what kind of things might be a solution in the long run?
So we've recognized that because pain is a subjective experience, there is no way to objectively assess pain. But what we recognize that with neuropathic pain, because the underlying cause is so not known, you're not sure if you should target the underlying inflammation, if you should target the underlying nerve that is damaged. One becomes confused as to which drug to actually administer to patients. So far, what we recognize is that antidepressants work, because sadly, the same seat of depression is the same seat that is affected by multiple sclerosis. So antidepressants work. We've recognized that medication that support nerve stimulation works, surgical operation works, but really there is a paucity of information that with regards to neuropathic pain research that will help us to guide therapeutics. But at the end of the day, the limitation will always be that one, it's an area that is under-researched, and two, no one can really objectively qualify pain.
Okay, thank you very much.
Brilliant, thank you. Tony or Mary, any questions for Trisha?
Can I ask Trisha, how important is talking about pain? You gave a very sort of vivid description of the sort of vast unquantifiable area, but is giving patients an opportunity to talk, is that significant?
Yes, an excellent question. And what we've recognized so far is that talk therapy helps a portion of MS patients. And really and truly, I want to underscore, it is really not the underlying pain that is being ameliorated by the talk. Is the fact that the associated complication that results from having chronic pain, it provides a way of reprieve. So the depression, the fatigue, the loneliness, that level of solitude is ameliorated. And if you can rehabilitate those patients whom you cannot treat for a lifetime in terms of eliminating their pain, and you can effectively shuttle them out into social activities, the mental distraction will help them to combat some of the offset or the bad effects of neuropathic pain.
That's very clear. Thank you, Trisha.
Brilliant, thank you. Tony, did you have any final questions for Trisha?
I was hoping to ask about a bit more about the sort of objectivity, subjectivity of it. So you must be sort of gathering data from your patients. What else can you do to try and gather data that is in some way comparable or objective?
Okay, so one of the ways that we in terms of objectifying pain measurement in multiple sclerosis is that we are trying to make that pain assessment scale more detracting from giving pain a number, but rather associating with emotions. So instead of saying, tell me your pain from 1 to 10, we will say, How does this affect your level of activity per day? How does this affect even your libido? How does this affect your ability to socialize with family and members? Does it make you more recluse? Do you normally have pleasures in things that you once had pleasure doing? So really and truly, in order to objectify pain, we're looking at not only a single dimensional assessment in terms of a facies scale or a number scale, but we are now taking on a multidimensional approach. The Leeds group have even come up with a scoring system to assess pain based on patients' the quantification in terms of number, how their level of activities are affected, how their sexual lives are affected, and other parameters. So it's a matter of incorporating all of those things that makes people intrinsically human into a scaling system.
Brilliant. Thank you very much for that. So our final contestant of the night, you've now seen our 7 contestants, each of which are up for a place to get one of those 2 spots for the Famelab final, the UK final, where again, the contestants will get some training. So they'll have a masterclass weekend before going on to compete with another 3-minute talk in the UK final, where we're looking for just one person to go on to that international final to represent the UK against a whole host of other countries. So I hope very much that you've enjoyed the talks so far. Stay with us because we're going to be finding out shortly who is going through to that UK final. And as I said, there's going to be a bit of a quiz. The next thing that happens is our judges are going to have to leave us for a little while, and they will be joining a separate call to discuss those 3 characteristics, that content, the clarity, and charisma, to decide which 2 contestants will be going through, and we'll also have a runner-up as well. So judges have a very difficult decision to make. Some people have already made that decision. We've got a comment from Sarah, who has a suspiciously similar name to Anna, and says, Anna, you're the best. I'm so proud. Lots of little clapping emojis and lots of little balloon images. So unfortunately, it's not Sarah who's making the decision, but you've certainly got a fan out there, Anna. So the next thing we're going to do is we are going to say goodbye to our judges. So let's wave bye-bye to the judges. And judges, you go off and disappear. Come back and see us in about 10 or 15 minutes with some decisions, please.
Bye.
And off they go. So our judges are away now, and it's just us. So the judges have a difficult decision to make. They'll be joining us again, hopefully in about 10 to 15 minutes with the results. But while we're with us and while we're in this science mood, I'm going to kind of give you a little bit of a quiz, test your science knowledge. To take part in this, you're going to need 2 things. You're going to need this feed because you're going to want to be able to see the questions that I'm going to put up on the screen, but also you're going to need some sort of second device or second window so that you can answer the questions that I put up on the screen. So in a second, I want you to have me up on your screen somewhere, maybe use your phone or a separate window. And in that separate window, I want you to go to kahoot.it. So you can Google the word Kahoot and click on the first link and it will pop up, or go straight to kahoot.it in your browser. I'm going to bring it up on screen so that you know what I'm talking about in a second. There's me, double me. Here we go. So kahoot.it, can you see that? Is that mirroring? Is that all okay? You can see it? Good, you can see it. So go to kahoot.it if you could, and the pin number for the game is up on the screen. I'll just get rid of the Famelab logo now and the Cheltenham logo so that we can see what we're talking about. So go to kahoot.it or click Kahoot in that Google link. And when you're there, I want you to enter in the game PIN number. And this is on your second device or your second window. The game PIN number, which will allow you to answer questions on screen, is 37107. Enter in that PIN number, and then you'll get to assign yourself a nickname. This will allow you to answer the questions that I'm going to put up on screen. The faster you answer, assuming you're correct, the more points that are available. Now, if you are playing on YouTube in particular, if you're watching this feed on YouTube, there's a little bit of a delay. There's a 6-second delay. So don't go by the counter, go by my cue. I'm going to say when I'm going to end the question. So if you're watching on YouTube, listening very carefully to when that time limit cuts off for you. So we've already got lots of people playing. That's brilliant. 22 players already in the room, 23. But again, to join in with this general science quiz, there is nothing needed installed. We're not asking for any personal information. We just want you to go to kahoot.it on your phone or in a second window, which is going to turn that window or that phone into a voting handset, and you'll be able to answer the questions that appear on screen. Excellent. We've got 30 players already joined us. We're going to, I wonder if I can do this, if this is going to work. No. Try and make this window as big as I can so you can see what we're doing. And move up a little bit. Is that working? Okay, there we go. Hopefully you can see what is on the screen now. The questions will appear there. And you'll answer. They're all multiple choice. Grab your phone and join in as we launch the Famelab Kahoot quiz. So here we go. First question is going to appear up on screen pretty soon. The pin number is staying on the bottom of the screen throughout. I'll move myself into a more convenient position. So first question is about element association. Which element is associated with this picture? Is it tin, copper, mercury, or gold? Once you think you've got an answer, as the picture reveals itself, click that answer as quickly as you can. So which element is associated with the image that is being unveiled slowly? You've got just a few seconds to answer. So we've got most people already answering. Is it tin? Is it copper? Is it mercury or gold? Which of the elements do you think has some sort of an association with this image? Your time is now up. I know it says there's time remaining, but the time has now stopped. Make sure you have clicked on all your answers. So stop answering now, and we're going to see how we got on.
I'm not getting on.
You're not getting on. Lots of people have answered,
I'm not,
though.
Yeah, no, I'm not seeing your... I'm on Kahoot. I've signed in, but I can't see what you're saying.
Sorry, Trisha, you need to open up Zoom as well. So you need to be in the Zoom call, and you need to have two windows at the same time. So if you're on one device, I realize that might be a little bit tricky.
Yeah.
Okay, some of us have answered. We've got 27 people answered correctly. That was Freddie Mercury in the picture. So we're going for a bit of cryptic element hunting here. 27 people got that right. Well done. That was the easy one. That was the easy one. A groovy elephant in the lead. Let's move on to our next question. And again, another element association. Which element fits with this picture? Is it palladium? Is it tungsten? Is it cesium? Or is it oxygen? Which element has a connection to this image? Answer as quickly as you can because the time will run out. Palladium, tungsten, cesium, or oxygen. What is the elemental connection to this picture that is unveiling itself? Again, most of the answers are already in, but the picture has now fully revealed itself and the question is closed, so no more answers. Make sure you get your answers in nice and quick, and we're going to find out the answer for this question. Lots of you getting that as well. I'm making them too easy. Trickier ones are coming up. That was London Palladium. So the elemental connection there, palladium. Next question is another elemental association, and Focus Piranha has now jumped up to the top spot. Answering as quickly as you can. Another element association. Which element fits with this picture? Is it magnesium? Is it lead? Is it arsenic? Or is it silver? What is the elemental connection to this picture that is revealing itself? The answers have started coming in. What do you think? Is it magnesium? Hit red. Is it lead? Hit blue. Yellow if you think it's arsenic, or green if you think the elemental connection is silver. Again, as a picture, once the picture gets fully revealed, I'm going to stop the answers coming in. So answer right now. And the answers are now closed. We're going to find out if you got this one. So what is the elemental connection with this picture? Have we got this? Oh, a bit more of a mixed response there. That was a still from the Cary Grant film Arsenic and Old Lace. So the elemental connection there was with arsenic. 10 of you getting that right, just 10 this time. Is Focus Piranha still in the lead? Let's find out. No, Focus Piranha's lost their space to the Expert Lima. Well, Expert Lemming, sorry. Well done if Expert Lemming is you. On to question 4 now. Last of the elemental associations. Which element fits with this picture that's unveiling itself? Is this related to krypton? Is it related to seborgium? Is it related to tellurium? Or is it related to neodymium? What do you think? The picture that's unveiling itself, what is the elemental connection? Answer as quickly as you can to get the most points. Once the picture's unveiled itself, I'll close the question, so make sure you're getting your answers in. Most of us have already answered. Well done. And we're going to find out the elemental connection in just a few seconds. So did you get this right? What do we think this was showing us? Well done. It was the planet Krypton, where Superman comes from. Krypton, the element, Krypton, the planet. The elemental connection there was Krypton. Who is in the lead now? Expert Lemming, knowing their Superman. Let's go on to question 5 now. Which bin should you put this biodegradable cup in? Click red if you think that should go into food waste. Once you're finished with it, should it go into mixed recycling? It is plastic. Is it a non-recyclable? So does it go into landfill? It's dealt with there. Or does it go into paper recycling? So a biodegradable plastic cup, which bin would you put that in? Food waste, red. Mixed recycling, blue. And well, all the answers came in really quickly, so the question has closed. Food waste is the correct answer. So actually most of us getting that wrong. A biodegradable plastic cup needs to be in a biodegradable compost environment. So if you have those plastic cups at home, they actually go in the food waste. If you send them to plastics recycling, they won't be recycled and they potentially contaminate the plastic there. So those biodegradable cups confuse a lot of people. But they did not confuse the fuzzy echidna. Fuzzy echidna. Don't think they are fuzzy, but everything's cute that's fuzzy. Question 6 of 12. Nobel Prize winners. I want you to try and put these in order from earliest to most recent. So you're going to have to reorder these. Who got their Nobel Prize first and who got their Nobel Prize last? We've got Marie Skłodowska-Curie, we've got Dorothy Hodgkins, we've got Albert Einstein, and we've got Wolfgang Pauli. Who got their Nobel Prize first? And order them to who got their Nobel Prize last as quickly as you can, because again, I'm going to close the voting a little bit early. So get those answers sorted. The earliest person to get a Nobel Prize and the most recent person to get a Nobel Prize. So first of all, you're starting with the person who got their Nobel Prize first. Who do you think it was? Marie Skłodowska-Curie, Dorothy Hodgkins, Albert Einstein, or Wolfgang Pauli? Who got theirs first? And the answers are now closed. I'm going to move on to the next question. Let's see how we did. Did you sort it out? The correct order was Marie Curie, Albert Einstein, Wolfgang Pauli, and the most recent was chemist Dorothy Hodgkins to win their Nobel Prize. So did we do well in that? Who is expert? Fuzzy Echidna, knowing their Nobel Prizes, well done. But Speedy Turtle climbing up the ranks there to be second place. Question 7 of 12 now. In 2016, the World Wildlife Fund gave a nickname to this recently discovered newt. But what is the nickname that they gave it? Is this a Jabba newt? Is it a Charmander newt? Is it a Targaryen newt? Or is it a Klingon newt? What do we think? Is it related to Star Wars, Pokemon, Game of Thrones, or Star Trek? Jabba, Charmander, Targaryen, or Klingon? What did the WWF call this newt? We've all answered. Very few of you getting that Star Trek connection. It's me. I'm asking the questions. I'm wearing a Star Trek t-shirt. Obviously, the answer is Star Trek. There was clues in the picture. So Klingon, because it's got the ridged forehead. It looks like a Klingon, but it's a newt. So WWF calling that the Klingon newt. Few more questions while the judges are out making their decision. Fuzzy Echidna still in the lead. Well done, Fuzzy Echidna. Speedy Turtle still in second place. Question 8 of 12. Tim Peake was the second person to wear a British flag in space, but who was the first? Red, if you think it was Nicholas Patrick. Blue, if you think physicist Sally Ride was the first person. Was it chemist Helen Sharman? Click yellow. Or was it pilot James Lovell? Click green. Who was the first person to wear the Union Jack in space? And love is knowing that. Excellent. Helen Sharman, another fantastic science communicator, doesn't do terrible many events, but she has done Cheltenham Science Festival, who organise Famelab. Did Fuzzy Echidna know that? They did. Oh, Fuzzy Echidna, very impressive. Knowing Duck, though, has now gone into second place, knocking that Speedy Turtle out of that second place spot. A few more questions still to go. Question 9 of 12. Answer as quickly as you can. What kind of ant is this? Is it a light farmer ant? Click red. Is it a docile orange ant? Click blue. If you think that is a raspberry crazy ant, click yellow. Or if that is definitely a fast brown ant, hit green. A light farmer ant, a docile orange ant, a raspberry crazy ant, or a fast brown ant. What is that? If you don't know, have a guess. The faster you answer, the more points there are available. Most of us have answered, so I'm going to close the voting in just a second, get those final answers in. And the correct answer is... It's the raspberry crazy ant. Why the raspberry crazy ant? It was discovered by a pest exterminator called Tom Raspberry. So he got to give his name to the ant, and it was called a crazy ant because it moves in an erratic way. So the raspberry crazy ant, named after the pesticide worker who discovered it and its erratic movements. The other three, entirely fictitious, made-up names by me. And that tricked most of you. But did it trick Fuzzy Echidna? You know everything. I'm really, really impressed. Is Fuzzy Echidna me? Because I set these questions. Nimble Shark now in second place as well, with a sturdy owl, a sturdy third. Couple more questions to go before hopefully our judges will be back. In 1963, the Memba effect was discovered, but what does it describe? Is it telling us that water can freeze faster than cold water? So that's warm water freezing faster than cold water. Does it tell us that ice is less transparent than water? Is the Memba effect the thing that tells us that water forms a perfectly round sphere in zero gravity, or that water has no taste to humans? So what do you think? The Memba effect discovered in 1963. Does warm water freeze faster, that ice is less transparent, that water forms a perfect sphere in space, or that water has no taste to humans? What do you think? And we're going to close the voting now. So final answers in, and we'll find out if we knew that. Let's see the answer, and it's that warm water can freeze faster than cold water. It's quite complex. They're not entirely sure exactly how it happens. Some people have thought that maybe the warm water forms a better thermal conductivity with a cold surface, and there's lots of different competing theories. But in 1963, they discovered that hot water will freeze faster than cold water, which doesn't really make sense, but it's true. Next question. Also, I see a little notification that our judges have made a decision and should be back imminently. But we have just a couple more questions. In which Sturdy Owl will attempt to knock that Fuzzy Echidna off the top spot. And Knowing Duck champing away at third place there. Question 11 of 12. Just two more to go now. True or false? Milk is acidic. Is that true or is that false? It's a common thing. Everyone said, you eat spicy food, you have some milk. Is that true? Is that absolute common sense? Or is it an urban legend? Which is it? True or false? Milk is acidic. What do you think? And again, final answers coming in. I'm going to close the voting now. So hopefully you've got your answer in. And let's find out. True or false? It's true. Milk is acidic. If you have kind of stomach acid problems, drinking milk is just adding more acid to the fire. Some reason we think that milk is alkali, but it's not. It's slightly acidic. So best not to drink it if you've got heartburn. Last question. Oh, Fuzzy Echidna, you're gone. Sturdy Owl knocking you to that top spot. Fuzzy Echidna now in second place. An agile gazelle leaping forward like an agile gazelle into third place. But we have one question left to go. And we're going to find out who is the winner. Which of these is regularly made from recycled plastic bottles? So these PET plastic bottles that are regularly recycled, what do they do with them when they're recycled? Do they make clamshell packaging, the thing that you get fish and chips in? Do they make carpets out of them? The soles of your shoes, is that where reused bottles go? Or does it make those little packing peanuts that you get inside new TVs and stuff? So what do you think? When a plastic bottle ends its life and it's recycled into another material, is it making clamshell packaging? Is it making a carpet? Is it making the soles of your shoes? Or does it go into making packing peanuts? What do you think? I'll give you 5 more seconds to answer. And let's find out. So recycled bottles end up making carpets, as a lot of you knew. So that's where a lot of bottles end up. We don't always reuse them to make more plastic bottles. Sometimes they end up in our homes in the form of carpets, making those little plastic bottles into those thin plastic threads. So we will see who has won our quiz, and then we're going to go back in to find out who has won the Scottish final of Famelab. So in third place, we've got Knowing Duck. Excellent. Sturdy Owl at second place. And who is in that top spot? It has to be. It's Fuzzy Echidna. Fuzzy Echidna. Well done. Fuzzy Echidna, are you one of the contestants? Are you in a room? Where are you watching? Give us a wave. Say who you are. Fuzzy Echidna. I'd love to know. So well done. 9 of the 12 questions correct. Hopefully you all find out how good or how badly you've done in that little quiz. Thank you for joining me, all those people who did. I hope you enjoyed that. But that is the quiz part over, and we're going to go back in to our Famelab call now to hopefully hear from our judges. So my first question is, are the judges back with us? They are. Hello, judges. Let's go to the room. Let's give the judges a wave. Welcome back, judges. So a difficult decision, no doubt, has been made. My first question, let's tease this out a little bit longer, X Factor styly. Maybe one of the judges could tell us a little bit about what they thought of the competition and the standard this year. Easy decision, tough decision. Judges, what did you think, first of all?
So I'll pick up that point. It has been an absolutely terrific evening. Each presentation was of high quality. It was very engaging. It was highly informed. Each individual, the piece that they presented, would stand up in any of these competitions. And my final point is, I come from the other side of the fence. I'm arts, I'm humanities, and each presentation I got it. And each presentation, I feel really confident. I could have sat down and had a conversation. So for me, all the things that Jamie talked about, charisma, content clarity, every single one of these presentations did it. And I have learned lots and lots of things that, had it not been presented that way, maybe I wouldn't have bothered. So we were absolutely knocked out by what we saw. And I'll leave it to my colleagues now to move into the very difficult decision we made. But it is no cliche to say that each presentation tonight was excellent.
Brilliant. Thank you, Mary. Did any of the other judges want to say anything else, or do you want to dive straight in to our announcement? Judges, are you there? We seem to have lost Tony. We've got Suzanne.
Oh no, poor Tony. I mean, just to stall for time. I mean, yeah, it was very much like, if the people we have picked weren't here tonight, we could have picked another 3 excellent winners. Everyone was top quality.
Yeah.
And really just, And also fair dues, because when you signed up for this process, when you went through your heats, it was an entirely different world, and you weren't expecting to be doing this in your bedrooms and front rooms or in front of a Famelab space sign.
Yeah, absolutely.
So, I mean, kudos. I mean, just turning up tonight and doing it is seriously well done to all of you. And of course, well done to Jamie for actually pulling all of this together
Yep.
in
Again,
a
yeah.
global pandemic. So
He's the
Thank
go-to
you.
guy, yeah.
it's his setup must be pretty spectacular over there. Have we got Tony yet?
We don't seem to have Tony. I've brought up gallery view, so just to let you know, you're all now broadcasting, so everyone can see everyone. Tony's screen is blank and his mic is muted, so I think we have lost Tony. So maybe Suzanne or Mary, maybe you'd be able to fill in for Tony and give us some results. So again, just to let people at home know, we're looking for 2 joint winners who will both go forward. They will get some expert training over the course of a weekend, and they will compete in the UK final of Famelab. Suzanne or Mary, we've got 2 winners and 1 runner-up. Who are they going to be?
So could I defer to Suzanne, because I think she actually wrote down the names, and I don't want to announce the wrong names because that would be a horrible end. So I'll pass to my colleague Suzanne to give you the names we finally selected, if that's okay, Suzanne.
Yeah, that's fine. We want to avoid any Oscar-worthy gaffes.
Oh, you read my mind, yes.
So we'll start with our runner-up, who, again, it was a very tough decision, and we thought this person really took something that's quite complex, and not just in terms of the subject matter, but also in terms of the societal implications, and made it quite understandable, and had a very kind of calm, engaging presence. That
absolutely handled questions beautifully.
Very much so. So our runner-up is Carolyn with her neural nets.
Well done. So Carolyn, well done, our runner-up. So we've got 2 more announcements to come now. So back to Suzanne. Our Famelab winners going through to the UK final of Famelab are...
So our first winner was somebody who, again, incredibly charismatic, managed to sum up their research in a single 4-letter word, and it is Trisha.
Absolutely. No, fantastic, yeah.
Well done. So Trisha
Very
Palmer,
well
our
done.
first person through to the UK final. Congratulations, Trisha. You obviously look absolutely delighted, so well done. Suzanne, you still have 1 more happy announcement to give us.
I'm just drawing this out for all these poor people. And our second winner, again, incredible storyteller, really took us on a journey. We learned things and we didn't feel like we were learning at the time. It was just so much fun, is Elizabeth and her rock pools. So congratulations.
And an incredibly approachable performance, making signs feel I had my wellies on here at home. If I'm going to go and put them on later.
Excellent. Oh, it all went so smoothly. I'm going to move. I've lost everything. Where am I? Let's move on to speaker view. I don't know. Where have I gone?
Have we lost Jamie?
2 seconds. Can you hear me?
We can hear you.
Oh, well, that's good.
It's so fun. And we can see each other.
I was trying to be fancy. And now you can't see me. And hang on, there we go. I can do this. It's all gone so smoothly.
Yeah, you're doing fine.
2 seconds. I'm going to be with you in a second. There we go. We'll go with this.
Yay.
Can you see me now? Excellent. Backup camera. Always have a backup. So well done to our 2 winners and our runner-up, but also a massive congratulations to all the contestants who really a fantastic performance tonight in very difficult circumstances as well. This is not an easy thing to do in the best of circumstances, all the more difficult to do it online. So a huge round of applause, I think, for every one of the contestants. Well done.
Fantastic.
And just to say thank you very much to our contestants, to Cheltenham Science Festival for organising this event. We'll look forward to the UK final and the international final. Thank you very much to Suzanne, to Mary, and to Tony, our wonderful judges. That was not an easy decision to make, and it was a joy to host you on this platform. Thank you as well to everyone who watched at home, who joined in at home. I hope you had a fantastic time, and follow Famelab on Twitter so you can watch the progress of the Scottish final. But bang on time, I want to say goodbye. Thank you to everyone for joining us, and I'll see you all again. Bye-bye.
Goodbye.
Bye-bye.
Bye.